Welcome to Học Tiếng Anh với Joseph, where we build vocabulary and practice pronunciation through reading. Continuing with The Cat in the Box, a history of science in 100 experiments. Today we will read story number 82. Chúng ta sẽ đọc chuyện số 82, A Blend of DNA. Even in 1951, in spite of the work of Oswald Avery and his colleagues, it was still widely thought that genetic information was carried by proteins, not by DNA. But then came the experiment that persuaded even the doubters that DNA is the life molecule. The experimental steps along the road that led to an understanding of DNA used a succession of smaller and more rapidly reproducing organisms. Gregor Mendel worked with peas, Thomas Hunt Morgan worked with fruit flies, and Avery's team worked with bacteria. The final step was taken using the smallest entities that carry genetic material, viruses. Viruses are little more than bags of protein, far smaller than a bacterium, filled with genetic material. When a virus attacks a cell, it injects this genetic material into the cell, where it hijacks the machinery of the cell and uses it to make copies of the virus out of the chemical material inside the cell. Then the cell busts open, releasing the copies of the virus to repeat the process. At the beginning of the 1950s, Alfred Hershey and Martha Chase, working at the Cold Spring Harbor Laboratory, developed a neat experiment that showed definitively that it was DNA that carried these instructions into the cell being attacked. They worked with viruses known as bacteriophages, sometimes shortened to phages, from the Greek phagos, to devour, because they eat bacteria. The idea behind the experiment was based on growing them in a medium that contained radioactive isotopes of either phosphorus, phosphorus-32, or sulfur, sulfur-35, and then using the radioactive phages to attack a colony of non-radioactive bacteria. The sulfur and phosphorus isotopes have different and distinctive radioactive signatures, and they can be traced through the cycle of infection and reproduction from one generation of phage to the next. Bacteria were grown in colonies laced with one or other of the radioactive isotopes before phage was added. The phages in the next generation took up the radioactive material and were then used to infect non-radioactive bacteria. The point of all this is that phosphorus is present in DNA but not in protein, while sulfur is present in protein but not in DNA. Everywhere the team detected phosphorus, they would be tracing the path of DNA, while everywhere they detected sulfur, they would be tracing the path of protein. The snag was, after the radioactive phages had done their work in a culture of bacteria, what was left was a mass of cells filled to busting point with new viruses but with discarded phage husks still attached to the hijacked bacterial cells. The culture still contained both kinds of radioactive isotope. Somehow Hershey and Chase had to separate out the leftover debris from the original generation of phage from the new viruses manufactured inside the bacterium. The difficulty was solved when a colleague lent them an ordinary kitchen utensil known as the warring blender. On a low setting, the blender provided just enough agitation to shake the empty phage bags loose from the cells they had infected, without smoothing everything out into an amorphous goo. After this agitation, the mixture was whirled in a centrifuge, where the bacterial cell full of new virus fell to the bottom and could be extracted, while the husks of the old phage were left behind. When the two components were analyzed, the results were persuasive. Radioactively labeled DNA was found in the cells, in other words, in the new generation of viruses, while radioactively labeled protein was found in the leftover husks. It was DNA, not protein, that was passed from one generation to the next. Although in the scientific paper announcing the results, the team cautiously concluded only that this protein probably has no function in the growth of intracellular phage. The DNA has some function. The success of this superficially simple experiment owed a great deal to the expertise of Martha Chase. Although officially she was only the assistant to Alfred Hershey. Václav Zimowski, another Cold Spring Harbor biologist, later recalled, experimentally, she contributed very much. The laboratory of Alfred Hershey was very unusual. At that time there were just the two of them, and when you entered the laboratory, there was absolute silence, and just Al directing the experiments by pointing with his finger to Martha, always with the minimum of words. She was perfectly fitted to work with Hershey. It was now clear that the protein provided the structural material in the bacteriophage, while the DNA carried the genetic information. The results were published in 1952, and the work became known as the warring blender experiment. After this, hardly any biologist believed that the genetic material could be anything but DNA, and the stage was set for the investigations that would reveal the structure of DNA itself. Let's go to the vocabulary section. In spite of, persuaded, doubters, succession, viruses, inject, hijack, bust open, bacteriophage, phages, distinctive, detected, detection, husks, debris, agitate, agitation, amorphous goo, intercellular phage, superficially, Thank you so much for your time, and as usual, I will see you in the next story. Bye for now.